Author Sharon Mollum's grandfather was diagnosed with Alzheimer's at age 71, but strangely enough felt physically satisfied when giving blood. There had to be a link between the disorder and such a personality, and Mollum set out to find it. It was not long until Mollum himself was exposed to the genetic anomaly hemochromatosis, and he began his search for a connection between unfavorable traits and their continuing role in society despite natural selection. In doing so, the author provides an explanation for the continuing prevalence of certain fatal illnesses that help us live tomorrow but may kill us 40 years from now.
Hemochromatosis disrupts the way we metabolize iron. As exemplified by the former condition and symptoms of athlete Aaron Gordon, it resulted as a cure for more severe illnesses like the bubonic plague. Similarly, cystic fibrosis arose in humans as a means to combat tuberculosis. Mollum then demonstrates how bloodletting has been employed as a way to cure the ill effects of hemochromatosis. Only through employing this method did athlete Gordon free himself from his death sentence. Without a doubt, bad mutations which lead to fatal diseases evolved due to other diseases with longer-lasting consequences.
Humanity has one perceived flaw: mortality. Alcor Life Extension Cryonics Lab, however, hopes that future medical science can help restore the dead to life or even keep them immortal. This will never happen. Human tissue contains water, which when frozen expands into crystals that may cut blood cells. But despite our inability to survive a deep freeze, we have still evolved an adaptation to protect against the cold. Constriction of the extremities is a way to drive blood towards our torso and keep the vital organs at a safe temperature. Along with shivering and the use of brown fat, the body also has another response to the cold: cold diuresis, or the need to pee. By eliminating water like shrunken grapes, there are fewer ice crystals and thus they cannot damage the capillaries.
Every illness has played a role in our survival and ultimate evolution. Cholesterol is no different. The production of vitamin D has relied on the conversion of cholesterol in response to ultraviolet B sunlight. Vitamin D is critical in ensuring healthy bone growth in children and maintenance in adults. It is also true, however, that in regions with little exposure to sunlight, cholesterol may pose a problem. With a lack of necessary sunlight, vitamin D cannot be produced as proficiently, and cholesterol begins to accumulate in the body, putting people at greater risk for heart disease and stroke. It seems that Newton was correct when he stated that all actions have a reaction.
Evolutionary adaptations in humans have indeed stemmed due to fatal diseases. Favism, for example, is an inherited enzyme deficiency that results in rapid, severe anemia if fava beans are consumed, caused by a lack of sufficient amounts of an enzyme known as G6PD. Along with free radicals, favism is most common in places where fava bean farms are located, showing that humans have evolved with a genetic mutation that is only like to cause problems when they eat fava beans. Malaria is also a common threat there, and G6PD deficiency protects against it, since the disease can only thrive on normal red blood cells, causing a less hospitable environment for malaria to occur. When someone with a mild or partial deficiency in G6PD eats fava beans, the parasite can no longer survive and malaria is eliminated.
When it comes down to it, every living creature has two goals: survival and reproduction. Malaria does this by manipulating its human host by weakening us, so that we are an easy target for mosquitoes. Mosquitoes bite the infected humans and then carry the malaria-causing protozoa to other people to infect. If every patient with malaria was covered and away from mosquitoes, then the malaria-causing protozoa could be placed under an evolutionary pressure in which the person would not be debilitated. Their mobility would actually cause the person to spread the protozoa even more. So once we have an idea of how we want something to evolve for our benefit, then we can adjust the environment so that the organism can change according to our expectations, but in a way that will benefit both us and the parent.
Just like our fingerprints, our genetic information makes or breaks us as unique individuals. But is our DNA strictly limited to inheritance? Throughout the past century, recent evidence has shown that retroviruses have played a significant role in shaping us as human beings from the very moment of humanity's existence. Retroviruses are a type of viruses composed of RNA. Using an enzyme known as reverse transcriptase, they transcribe themselves from RNA into DNA and can thereby paste themselves into an organism's genetic line of code. An organism's offspring is thus born with the virus DNA permanently encoded in its DNA, but natural selection decides that the virus remains a permanent part of the organism's genetic pool. Thus, viruses have become a part of us, and their vast library of mutations allow us to employ them in our quest to evolve.
Mutations aren't the only proponents of sudden phenotypic change. The term epigenetics was coined in the 1940s. It erased the idea that our DNA was immutable. Now there was a whole new layer to consider, in which the DNA code itself did not mutate, but the traits could. Methylation is a result of epigenetics, during which methyl groups bind to certain genes and suppress their expression without affecting the actual genetic code. This discovery was first made following a study of agouti mice and the possible implications of vitamin supplements provided to them. When pregnant mothers were fed the vitamin supplements, the agouti gene was methylated, and thus their DNA was no longer expressed. For the first time, scientists could prove the correlation between the mother's nutritional supplementation and the offspring's gene expression.
Humans and marine mammals have a connection only recently discovered. They all have fat directly attached to the skin. The only way humans could share this trait with an aquatic mammal would be if they were an aquatic ape. The aquatic ape hypothesis states that pre-human ancestors lived near the water as they dived and caught food, learning to hold their breath underwater. Apes that were frequently in the water evolved towards bipedalism so that they could walk upright and move into deeper water, still maintaining breathing. This theory also explains why we lost our fur, just like other aquatic mammals. So that we could adapt to the water, we became more streamlined, and our downward-facing nostrils allowed us to dive. The aquatic theory also explains why we have fat attached to our skin. It allows us to float and flow smoothly through the water